um, may take this away.
All right. Uh, Hello, everybody. Welcome to our presentation. The topic that we chose was polypeptide and polyamino acid structural and functional materials. It's a bit of a mouthful. The instructions for this presentation were pretty broad, so it allowed us to pick a topic that was really broad and it gave us a lot of freedom. So I'm hoping you guys find it interesting and maybe learn something new. All right. Oh, I cannot change my slides. There we go. Before I start, I just want to say a short thing. Anyone who see me give a presentation before know my love-hate relationship with chemistry. It's interesting to learn about, but chemists like to speak in their own language all the time. So the way things are presented, it's explained in such a way that the first time I see it or hear it, I really don't fully understand it, even if I know the terminology. But to make things a lot easier, I always explain it like you're five, because then you'll understand it a lot better. And so it's a little everywhere at times, but I'm hoping it flows and I'm hoping it's easy to follow. So, over the last few weeks, the terms that we should be familiar with are proteins and peptides. And I hope we know at this point both are made from amino acids, with proteins being made with one or more long polypeptide chain, while peptides are classified as monopeptides, dipeptides, and oligopeptides. I hate using words to describe other words, but basically, peptides are just short chains of amino acids linked by peptide bonds, normally around 5 to 50 amino acids long. That's where the line is drawn between peptides and proteins, I guess, because once there's more than 50 amino acids, it's considered a protein. But proteins are generally greater than 100 amino acids long. Proteins also have defined 3D structure that determines function and activity, while peptides have no well defined 3D structure and no independent biological function. Now, with those definitions out of the way, we can move into the materials in question, which are polypeptides and polyamino acids. They sound like the exact same thing, but polypeptides are defined as long, continuous, unbranched polypeptide chains, peptide chains, and polyamino acids are defined in the literature as a polymer composed of amino acids as monomeric units, which basically just means it's a polymer with the monomers being uh, amino acids. And going back to polypeptides, polypeptides fall into the two broad categories of biological polymers and oligomers. Now, oligomers are defined as a polymer whose molecules consist of relatively few repeating units. The difference between an oligomer and a polymer is that an oligomer forms when a few monomers undergo polymerization, as opposed to a polymer, which forms when a large number of monomers undergo polymerization. An oligomer contains a small number of units, similar to how, like, a peptide contains a small amount of amino acids. And the other type of polypeptides are biopolymers, which are defined as naturally produced polypeptide chains. Examples of biological polymers include polynucleotides like DNA and RNA, and polysaccharides or unbranched or branched polymeric carbohydrates such as starch and cellulose, or proteins such as actin for muscles and collagen for connective tissues. And collagen is one of the most common biopolymers and is very abundant in mammals. And for that reason, we'll dive a little bit deeper into collagen. It's probably something people are at least slightly familiar with. Uh, Collagen is a major structural protein found in vertebrate tissues. Collagen is the most abundant protein in mammals, composing about 30% of the total protein mass. And there are 28 various types of collagen that have been discovered in humans, with every one of them being given a Roman numeral from 1 to 28. But lucky for us, we don't need to flex our knowledge of Roman numerals because collagen 1 makes up 90% of the collagen in the human body and is found in tendons, skin, cornea, bones, and lungs. Now that we've explained what collagen is, let's explain what collagen is on a deeper level. So, since it's a protein, it's obviously made out of amino acids, but that doesn't really cut it. The general amino acid sequence of collagen is glycine XY. And essentially, every third amino acid is glycine, but X and Y can be any amino acid residue. But the amino acids X and Y are most commonly proline and hydroxyproline. so that the collagen molecule can form a triple helix. Yeah, like I said, collagen is composed of three chains, which are then wound to form a triple helix. Because every third amino acid is glycine, it allows the chain to form a really, really tight configuration and can withstand massive amounts of stress. The triple helix collagen undergoes is particularly strong because the left-handed polypeptide chains twist to form a right-handed triple helix. Kind of similar to rope. I don't know if that's like a weird analogy, but every single tiny piece of rope in that twisted rope. Let's see if I can get the laser pointer going. Well, I don't know what I did. Oh god, I'm messing this up. There we go.
It's hard, eh?
It is very hard being on the other end.
There's you turned on your closed captioning. I think it's the button next to it. There's a little.
Oh, I've lost my mouse now. God, the pressure. Oh, there we go. Okay, perfect. Anyways, where was I? Oh yeah, my analogy to rope. Every single tiny piece of rope in that is twisted one way, and then the small strands are twisted in the opposite direction. It just strengthens the material a lot. All right. And I know we're all chemists here wondering what the chemical properties of these materials are, but I just wanted to highlight some of the physical properties that they have, and more specifically, the physical properties of the material catgut and its relation to collagen. Now, catgut is kind of a rather upfront name for a material, but it's essentially a natural biopolymer of collagen fibers that are found in the intestines of animals. The name suggests that it comes from the intestines of cats, but it's believed that cat was shortened from cattle, if anyone cares about the derivation of the word. And this whole year has been like a organic chem class, essentially. So you're probably wondering what's the synthesis of this material, but you can't even call it that. It's more of like an extraction, because cat gets prepared by washing an animal's small intestines, normally a sheep or a cow, and soaking it in a strong base to remove any soft tissue. And then the strands are twisted together multiple times, depending on the thickness required for the strand. And you're probably wondering, why do you need a thick strand or a thin strand? Because catgut strings have tons of applications. It was used in high performance tennis rackets. It was traditionally used to hang the heavy weights in grandfather clocks. And the use of catgut I personally use is the use of catgut in instrument strings. I don't know if there's any classical music fans in here, but I personally use catgut strings for my violin. It should be known I suck at violin and haven't played in 4 years, mostly because cheap strings will rack you up around like $125. But regardless, catgut strings are the perfect structural material for instruments such as violin and cello, because a cello can have like about 200 pounds of torque in the body of the instrument when the strings are stretched to the full tension. And they're strong as hell, so may as well use them. There's evidence of catgut being used way before the discovery of modern chemistry too. Like humans were, I know it's irrelevant and doesn't really matter, but humans were using woven intestines for a long time before instruments and long time before chemistry. And I'm not gonna lie, this is also irrelevant, but it's kind of too interesting to not include. Right around World War II, catgut was in big demand because there's another use for catgut that's really important. It's an absorbable surgical suture. So musicians had to switch to another big discovery at the time due to the limited amount of catgut, nylon. I believe nylon was like one of the first polymers ever discovered. And now it's more common for nylon 6 polymers to be used because they have the similar molecular properties to catgut. And nylon here, it's a polymer, so it has peptide bonds, but the big difference between nylon and catgut is that the fibers don't form a triple helix in nylon. They H bond and stack to form fibers. So they're still strong, it's just in a different way. So why did they use catgut as a suture and not nylon? It's because catgut has many uses in medicine because it can be broken down and absorbed by the body. It's commonly used as an absorbable surgical suture, but it can also be used for bone grafts, tissue regeneration, cosmetic surgeries, and cardiac application. The most common catgut suture is called chromic catgut, which is nearly identical to catgut strings used in instruments, but they're coated with chromium salts to preserve the tensile strength of the material until healing is achieved. By treating catgut with chromium salt solutions, it allows the catgut to resist the body's enzymes and slow the absorption process, allowing the suture to keep its tensile strength for a longer period of time. Catgut sutures that remain untreated are called catgut plain sutures. They're basically just used for like fast healing tissues or for like stubborn children who don't want to have sutures removed. So, as displayed, polypeptides and polyamino acids are great structural materials that can function as absorbable surgical suture very well. But chemists always want to attempt to create synthetic materials that can replicate the process natural materials can do, and sometimes make the material better. But the problem with catgut sutures is that they lose a lot of tensile strength before a wound is fully healed, around 7 to 10 days. So they're only used for fast healing tissues. The way to get around this is to create a new material that is absorbed slower, but also has the same strong tensile properties as biopolymers. So I was looking up some names of synthetic sutures, and it was really difficult because almost all of them are referred to by their branded copywritten name, and it was really hard to find out what they were made out of. Because you look up these materials, it just brings you to like a business website. But after some digging, I found that almost all of them are made from similar materials. The majority of the synthetic sutures are polymers with glycolic acid as its monomeric units to form polyglycolic acid, or PGA for short. Now, polyglycolic acid is a biopolymer, but it's not held together with peptide bonds, and it has no amino acids. So this material really has nothing to do with polypeptides or polyamino acids, but they are the synthetic version of the polypeptide biopolymers that occur naturally. So I figured I'd include their synthesis. So the most common method used to produce polyglycolic acid is the ring open polymerization of glycolide to polyglycolide. For the ring open polymerization to occur, an appropriate catalyst has to be used so that the reaction can occur in reasonable conditions. And a common catalyst used for industrial preparation of polyglycolic acid is tin-2 bis-2 ethyl hexanoate, commonly referred to as tin-2 octanoate or SN-oct2. You know how it goes. All right, so it was ridiculously hard to find anything related to the synthesis of these materials, but I found the predicted mechanism for the tin octanoate catalyzed ring opening polymerization of polyglycolide. God, it's a mouthful. And if this polymer chemistry makes sense to me, it should make sense to all of you. All right, so the tin catalyst in methanol, or the tin catalyst when in the methanol solvent, allows the methanol to coordinate to the tin center. And I'm not gonna lie, this mechanism is pretty messy to me, but I had to recreate it because it was even more messy when I found it. But it allows the glycolide to just hop right in and bond to the tin center. And it bumps in, and coordination's happening all over the place in this molecule. But the glycolide yoinks the oxygen from the methanol that was coordinated to it. And if you've managed to follow it that far, the oxygen on the glycolide ring here manages to scoop in and bond to the hydrogen and open to cause the ring open polymerization. Not sure if that explanation made too much sense, but in the end, you end up with the start of your polyglycolic acid chain, aka PGA, aka polyglycolic acid, polyglycolide. So if I lost you on the synthesis, all you really need to understand is by recreating many of these natural biopolymers and polypeptide chains, the chemical properties of these materials can be altered to be more favorable for certain situations. Traditional catgut loses most of its tensile strength within 7 to 10 days and is absorbed after about 60 days, while polyglycolic acid sutures can keep their tension for almost 2 weeks and are absorbed at about 90 days. So in conclusion, If you were to take any information from this presentation, I'd like you to know that polypeptides and polyamino acids can be useful structural and functional materials. The natural structure of the polypeptide chains in the protein collagen allow it to be obtained as strong fibers in the material catgut. And the strength of the triple helix between the polypeptide chains and collagen allow catgut to be used as a strong material for things like instrument strings, and its biological chemical properties allow it to be used as surgical suture. And the unique properties of these types of materials allow it to be used for many possible applications. And that's the end right there. I don't know if we should do questions now or if I should just hand it off to Luke.
So it's kind of like a two-parter, like Luke's doing a second part.
Yeah.
Is it connected or pretty loose?
It's connected to the materials. I think he's doing drug delivery applications.
Yeah, it's more applications.
I think we can hold questions, and maybe Luke, you can go, and then we can keep the questions for, throw them all out at once.
Sounds good. All right, everyone, see what's going on here? Okay,
Yeah, we can see your screen. Yeah,
perfect,
we can see your screen, and we can hear you, so.
Cool. So I'm just going to pick up a little bit where Michael left off here, and I'm going to go over a few applications. So I'm going to start off with drug delivery with polyamino acids. If my slide would like to change. Here we go. Okay, so I'm going to start off talking a bit about the research leading up to the use of polyamino acids in drug delivery. So proteins were once and still are being looked at for use in the medical field for drug delivery. As I'm sure most of you know by now, proteins are made up of a sequence of 20 different amino acids, the order of which and the structure determines its functionality. So, for example, here we have spider silk protein called spindroin, which we can see here in this picture, consists of different segments of amino acid blocks, mostly made up of alanine and glycine. So in this instance, the amino acids come together to form a strong structural protein. Another type of proteins are enzymes. These are the proteins mostly looked at for drug delivery, and these contain catalytic functions used to initiate chemical reactions in biological systems. And so, yeah, as I said, these are the types of proteins being investigated initially. So enzymes in particular were being looked at for drug delivery due to their ability to be easily tailored and in their biological function, just meaning that by altering the protein structure or by changing the amino acid sequence, you can easily target certain organs or cell types for drug delivery. Another reason enzymes were ideal for this drug delivery application was that they could form nice cradle-like structures in which therapeutic drugs could reside in here. So as in this picture here, it's a little bit of a cartoony diagram, but you can see just in the middle here, these yellow dots are supposed to be the payload, the medical payload, and it's encapsulated in this protein. And finally, they also have the ability to interact, fold, and transport DNA, which was another attractive feature for researchers. So despite the appeal towards using these enzymes for drug delivery, there were many perceived issues. The first one being which they realized that the engineering needed for the synthesis of these of many of these conceptualized proteins was beyond capabilities of modern technology. So the proteins, proteins in general, can get very large, and controlling every small detail of these proteins can be very difficult, and that was one of the reasons for this. So even in cases where synthesizing the proteins are possible, making it in a usable quantity is also difficult due to the fact that so many things can go wrong, and everything has to be done very carefully. Another issue that stems from the complexities of proteins is that the enzymes can interact with many different parts of biological systems, and controlling every interaction within a body or biological system is nearly impossible. So this leads to a degree of unpredictability that is not normally tolerated in medicine. So knowing all of these issues, there's been a push towards using polyamino acids as an alternative for protein drug delivery. So using polyamino acids instead of enzymes solves a lot of the big problems discussed previously. The first one being that amino acids allow simpler synthesis since they are made up of amino acids which are much simpler and whose interactions are better understood. These materials are still highly tailorable and can be altered depending on their desired function. And also in research where hydrophobic drugs are being used, and these amphiphilic copolymers, which I'll discuss in the next slide, have been shown to be particularly useful. So when designing polyamino acids for drug carrier synthesis, the chemistry of both the drug and the solvent are something we need to keep in mind. So since we're talking about drug delivery and biological processes, most of the medicines we're going to be use need to be water soluble. This is just simply because we are water-based creatures, and most mammals are. I guess all mammals are, and most systems in our body are hydrophilic, with the exceptions of fattier organs like brain tissues. So in order to make a polyamino acid, acid carriers that are water soluble, the outside portions of these molecules or carriers typically have to be hydrophilic or polar. So the way they did this in many examples that I was researching was by using these diblock copolymers, such as this example here with leucine and lysine. So each strand here you see sort of stemming out from the middle is the outside portion is a hydrophilic, is the hydrophilic leucine, and so it contains polar groups. Oops, sorry. Yeah, so it contains polar groups, giving it its hydrophilic nature. And so these more dense groups here towards the middle are the hydrophobic or non-polar portions of the copolymers. And so in that case, this is lysine. So as I mentioned in the previous slide, these were particularly useful for hydrophobic drugs. And that's just due to the fact that the inner portion of these structures are typically non-polar when dealing with these copolymers. So they're easily soluble, or yeah, they can easily attach themselves to the middle of these structures. So the last thing I'm going to discuss about drug carriers is their self-assembly. So the way in which polyamino acids self-assemble is dependent on the size and configuration of the copolymer parts. The drugs that are being transported by these carriers are either preloaded into the self-assembly so that they are designed to assemble within the nonpolar region of the carrier, or alternatively, they are designed to be just dropped into solution with the drug in solution as well, and to be uptaken in the aqueous media. So going back to the stuff I discussed in the last slide, the self-assembly of amphiphilic copolymers is driven by the hydrophobic portion of the copolymer grouping together and the positive interactions between the hydrophilic block and the solution. So I just had a little diagram here of just sort of the different structures that these polyamino acids can self-assemble into. This isn't necessarily, these aren't necessarily drug carriers, but just different examples of their self-assembly in general. So we have at the top here, these are the ones we were discussing previously, these block copolymers with the hydrophilic and hydrophobic portions. So you can see that these sort of form these, well, yeah, like in the previous example, these carrier structures where the hydrophilic portions are driven towards the outside where the aqueous solution is, and these hydrophobic monomers of the copolymer direct themselves inwards to reduce the repulsion felt by the solution. We have another sort of shape here, these graph copolymers. Sort of a similar concept here. Hydrophobic portions stay inwards while these hydrophilic parts dissolve in the solution. These make a little bit larger of vessels here. They also can form these solid nanospheres. And I guess these peptide amphiphils also can form these nanofibers. I didn't look too much into that, but that was just another example. All right, so my next portion of this presentation for applications is going to be discussing a bit of about biodegradable polyamino acids and their alternatives to petroleum-based materials. So I'm sure this isn't new to anybody, but humans use a lot of plastic. So every year, over 200 million tons of petroleum-based plastics are produced, while only 750,000 tons of bioplastics are produced. However, this number is increasing. 20 to 30% of all land mass is plastic, and only about 50% of all plastic that gets used gets recycled. This is especially problematic because of the long periods of time it takes for petroleum-based plastics to decompose, which is up to 450 years. So this picture here on the right is the Great Pacific Garbage Patch. What a name. It is located in the central North Pacific Ocean. It's just a stretch of ocean, 1.6 million square kilometers, which is just covered in garbage. And around half of that is all plastic. So obviously this is an issue we need to deal with. So most eco-friendly polymeric materials consist of bio-based polyesters due to their plasticity and workability. However, these types of polyesters lack toughness, which limits their applications. Another type of biomaterial that we talked a bit about, that was talked a bit along with polyamino acids, was polyamides, but they were considered a bit impractical for large-scale production due to their complicated synthesis. So polyamino acids are found in many structural materials in nature, as Michael did a good job of explaining. And so it's logical to think that scientists would try to mimic these properties of these materials. So one example of a naturally occurring polyamino acid, or I guess it's more, polyamino acid containing material was spider silk. So here's a closer example of spider silk. We have these alanine sort of threads grouped together, and these alanine strains are connected to these glycine-rich coils. It's not completely just glycine; there's also other amino acids as well, but they're glycine-rich, and these sort of act as springs when pulled apart. And you can see here in this diagram how when a tension is applied, these tend to stretch out, and so this is what continues to or what contributes to spider silk being one of the most strongest biologically produced materials. This other picture here just shows a couple of other examples. We have, again, spider silk being spooled onto this sort of coil here. We also have silkworm from here, and these blue mussels. The adhesive they use to stick to the seafloor is a polyamino acid based material. So in this slide, I'm just going to go over an example of a synthetic polyamino acid that was made by this group here. Here's everything about this paper, and if you guys want to look this up, I thought it was a pretty interesting paper. But anyway, basically what they did was they used polypeptide containing nylon, and they used polyamino acids containing nylon, and they used these between alanine monomers to see if it would induce thermoplasticity. So just meaning that it would have plastic-like properties, which could be melted, molded. So if you look at this picture here, you see two alanine groups on the ends, and in the middle there is a nylon molecule, and is just denoting its length. So they did, I think, 4 or 5 different lengths of the nylon portion in the middle, and just to see which one performed best for plasticity. I believe it was a nylon 3. So the method they used for synthesis was by chemoenzymatic polymerization. I'm going to discuss a couple different types of polymerization in the coming slides, but that was the one that I saw most. Anyway, so in the end, what they found in this study was that the alanine and nylon mixed polymer showed a high degree of plasticity and was able to be melted and molded much like regular petroleum-based plastics. And it also demonstrated good biodegradability with nylon 3 when tested with seawater. So yeah, as I said earlier, I'm just gonna finish up my part of the this presentation with a few examples of the different polymerizations I came across. So the most common type of polymerization I saw was previously mentioned chemoenzymatic polymerization. This was by far the most used method due to it being the most eco-friendly and because it gives a very high yield and stereospecific product. It is also very easily thermodynamically or kinetically controlled and operates under relatively mild conditions and a reasonably short time. The only drawback that I really saw concerning this method was that the molecular weight was a little bit hard to control. So different sized threads or polymers, it was a little hard to control the size that you were looking for. So this type of polymerization is relatively simple. It just sort of relies on an enzyme protease, also kind of shown as papain right here. That's just another sort of derivative of the enzyme protease, and all this does is pretty much just facilitate the peptide bond formation. So in these pictures here, you can kind of just see the addition of protease. It just facilitates the attachment of the ester to the amide. So yeah, this is the example from the last slide here. This whole molecule is sort of treated as a subunit, and when treated with the enzyme, reacts with another molecule of itself through the amine to the ester, and just then polymerizes. So as Michael already went over in a much better detail than I will, is the spring opening polymerization. So I'll just briefly go over these two. These were not, there was one after this one. These were not really too discussed that much, but I feel like they're worth mentioning. So another type of polymerization was ring opening. This method uses ester or amide containing cyclic structures, which are opened and form chain structures that connect to other cyclic esters or amides. So in this case, a Lewis base is typically used to initiate the polymerization, but cations can also be used, but are much less common. The driving force behind this polymerization is the release of ring strain from the opening of the ring, as you can imagine. So this is a very simplistic example here, obviously. This bond between the carbonyl and the oxygen is broken. This long chain is formed. This is a favorable procedure. It alleviates ring strain. And what can happen here is that this carbonyl will interact with another oxygen of another one of these monomers. And this oxygen will react with another carbonyl of another one of these monomers. And this sort of continues the polymerization. So the last type of polymerization I'm going to discuss is group transfer polymerization. This one was probably my favorite that I saw. I just thought it was interesting how these group transfers work. But this was developed by DuPont in the late 80s. And it was commonly used for the high level of control that you can get in terms of molecular weight of the product of polymerization, and these silyl ketene acetals are used as the reaction site, and they migrate to each newly added monomer. So this reaction is catalyzed by a Lewis base or nucleophile and induces a Michael addition, shown here in the second step. So we add the nucleophile. There's a Michael addition that occurs here. During the Michael addition, you can see that this silicon-containing group gets sort of transferred over to the newly added monomer, sort of corrects itself here, and we end up with the polymer ready to recombine with the catalyst and add another monomer to the unit. So yeah, so this silicon, these silyl ketene acetal is what is migrating. It gets passed on between each consecutive monomer. All right, so that is all I have for you guys today from me. Thank you guys all for listening, and Michael and I will take questions. Thank you.
Thanks, Luke. Again, good presentation. And I think you guys both covered the brief. Like, the idea is that's pretty broad. No concerns about this being out of scope. This is well within scope. So open the floor to questions from all the grad students.
Instead of questions, more of a little bit suggestion. I learned from some. It's about your presentation. Both viewers are pretty good. It's all quite good. It's just that for your slides, it would probably be a better idea if you put down less words in there, because when you show full sentences, people just start reading them. They just like people couldn't help, so that they wouldn't focus on what you're exactly saying, and you want people to focus on, well, what you're talking about instead of what you've written down 3 days ago. So instead of a full sentence, you can probably put down just some short words as, kind of key points, keywords for things you're about to talk. Yeah, it's just that.
Okay, feedback is appreciated. Me and Luke both are in Dr. Loeb's supermolecular chemistry class, so we're just kind of used to that presentation style where we were just reading slides basically.
Oh God.
Yeah.
Sorry. I wasn't meant to be unmuted. Okay. I take your point, Hazel. But I think, and I would say that, look, personally, I agree. I never put words on slides if I can avoid it, but that's neither here nor there for the content of the presentations. So I think that is something to consider as a style, and I think any presentation style that works for you is the right presentation style to use. I think trying to shoehorn yourself into somebody else's style is challenging. Personally, I'm completely on board with Hazel's comment as well, but that's not going into my evaluation at all. Anyone have any questions about the science? I think there are participation grades for the grad students for participating in the questioning period here. There's a lot of stuff to ask.
Can we actually go back to the tin mechanism? Because I totally got lost there. Yeah, it's from.
I think that was Michael's.
Michael's, yeah.
Yeah, that one's mine.
It was interesting. It just also was quite confusing.
It is admittedly very confusing.
I think the biggest challenge with this is the arrows cross in the lower left, and it does make it in the same color. It does make it a little bit difficult to understand the order of things there. Like, I get what you're doing. I think you have an arrow going to the carbon where I think it should be going from the tin oxygen bond to the oxygen carbon bond to generate a new oxygen carbon double bond.
Yes, that is something I messed up. Well, I'm not gonna lie, my ChemDraw expired, so I had to make this in Adobe Illustrator.
That is challenging. You should have free ChemDraw Prime from university. For the Software Depot, you can all download ChemDraw for free.
I wish I knew that at the time.
Okay, yeah, that would help. Sarah, okay, Hazel, if you want to ask your question.
Actually, I'm just a little bit confused with the very first structure. So, like the little thing in between two oxygens, that's supposed to be a resonant double bond.
Sorry, can you ask that again?
Sorry, the first structure, like the little thing in between the two oxygen on the upper half.
On the far left structure.
Starting material. Yeah, that one.
Hazel, you can actually annotate the screen as well if you want. Under View Options across the top, there's a button that says Annotate. And I'll bring up a little toolbar, and you can actually draw on the screen.
This one.
Oh, this. I'm pretty sure that's shared resonance between the oxygens.
Hey, that's a show resonance.
I think I actually forgot to put one below here, but that's what it showed in the paper that I found.
Oh, yeah.
That one's on me.
That's okay. I figure so. I just want to be sure.
Okay. Apologies for that. Any other questions about it?
Yeah, no, I just want to look at this again because it's definitely a very interesting mechanism, and I like, yeah, it's pretty cool.
So
Pretty well
what do
done.
you think are the key features of, so essentially, I guess this is a question for Michael, following up on sort of Hazel's thing here. This is essentially using tin as a Lewis acid. Do you agree?
God, you're testing my knowledge of definitions. Lewis acid. What does that do again?
This
Accepts
has is taking
electrons.
electrons.
It's like an H
Electrons
plus that
are-
isn't it's H plus that isn't H plus.
It's H plus that isn't H plus.
Anything that's not hydrogen that's acting as an acid. So the tin is accepting electrons from the methanol and also from the carbonyl, right?
Yeah.
So like the really the big thing that you're doing here is in your third top right structure thingy, that guy. Because you've activated the carbonyl of the glycolic anhydride to make it electrophilic, basically, so you can do the chemistry, right? So what do you think about, why do you think this tin thing is used instead of like other Lewis acids? Because you could use boron to do that, you could use silicon, you could use lithium, like almost anything would activate that, but there's something kind of cool about the way this tin works.
I feel like you know the answer, but I would probably say it's just able to allow this oxygen to attack it easier.
Okay, I think all the others would too. I don't think that's what's unique. I actually think tin's not great for that. There's better ones for that, but there's something else here that makes it go, oh, it just, this lithium wouldn't work, for example, for this.
I'm not too
You have
sure.
it drawn on your slide. You have everything drawn.
I have everything drawn.
Yeah.
Is it that you only it's a catalyst, so you probably only have to use like small amounts of it. I'm not sure if that's the answer you're looking
No,
for.
because all Lewis acids would be the same. I guess if I threw in lithium plus, I would be able to activate the glycolic acid as well, the glycolic anhydride as well. But is there?
I feel like you're handing me an answer here, but I just can't answer it right now.
Okay.
I understand that feeling, may I?
What you're doing here is you actually have this other six-membered ring formed because of the octanoate, because you're using what it's doing is it's using the octanoate ligand, like that's the acid ligands that are on the tin to begin with, right? R equals C8. It's irrelevant that it's C8. It doesn't matter. It's all about solubility for why they choose that particular ligand. But what you have here is the tin's acting as a Lewis acid, and then you've also activated the octanoate as an acid, right? You have a hydrogen binding to the methanol.
Yeah.
So the hydrogen's actually shifted off of methanol onto the ligand. Because what you've done is you've broken your both oxygen bonds to tin. One of those has been released to generate the carbonyl, right? In your second structure. And now that can grab the hydrogen from methanol. So now your methanol is basically a nucleophile, it's basically O minus. So you essentially have O minus and a positive charge in the same molecule right next to each other. And other Lewis acids don't have this right geometry. So there are other, like lithium is only monovalent. It can bridge things, it can be divalent, but it's uncommon. Whereas other bigger Lewis acids like iron or lanthanum don't have this. Do you know the shape of this tin? Like the thing you drew at the front. You've drawn it all on a plane.
Right here?
Yeah, you've drawn it all like in one plane. Is it all in one plane?
Yeah, I believe so.
Yeah. So that kind of crossed planar structure like that, tin is a big enough atom to accept that you can have these two four-membered rings fused to it, whereas not many other Lewis acids can do this. And what that allows is for that geometry to occur at the top. So you can sort of set up this acid base. It's doing both acid reactions and base reactions at the same time. And that's what's key, because that's what allows you to do the arrows you push in the bottom left structure, and allowing all that to happen. Because that oxygen of the glycolic anhydride is close enough to the hydrogen to take it. It doesn't look like it from your angle there, but it's just because you're drawing a three-dimensional object in 2D. while allowing a new double bond to form, and then you generate something which is so your bottom right structure. That's analogous to another structure you have here in the mechanism. Which one is it basically identical to?
Sorry, repeat the question again.
So that structure is analogous, it's identical to another structure you have here on the slide. Which one is it identical to?
Is it this one?
Yeah, right? So it's set up to do the next addition immediately of
Yeah.
the next unit. So this is how the polymer, because this is almost like the first step of the ring opening. And this is setting up the polymerization, because now it can add another equivalent of glycolic anhydride, and your residual or your growing chain is playing the role of methanol did in the original mechanism. So it's pretty cool. This is very careful catalyst design. It's a very simple catalyst, but very, very careful catalyst design. Brave of you to put it up.
Thank you.
Other questions? Okay, I'm really sorry. I guess I'm going to be asking a few more. So initially you were talking about glycine. I'll start with you, Michael. So you're talking about glycine making these triple helices and being really, really important for that.
Yes.
Why is glycine important for making a triple helix?
I know this one too. The triple helix, the glycine allows the triple helix to form because it's the smallest amino acid, so it allows it to meet in the center. And every once in a while, there's proline and hydroxyproline, so it allows it to coil into this big chain. And the glycine just allows it to like scrunch in further because it's so small, basically.
Yeah, I agree, completely agree. So like the one comment I have is I think I'm going to say I don't think you generated all these images yourself.
No.
So you don't have any references on your slides. So that's because the referencing is worth something in the marking scheme. So what I'm going to suggest is that Luke had the same thing, that both of you maybe add some reference to the slides and then submit those to me. I won't evaluate the references with the presented slides. I'll let you add them back in. I thought this was actually pretty cool. What do you think the role of hydroxyproline is? Why isn't it just all proline? Like hydroxyproline is not one of the canonical amino acids. So why do you think, because then you have to go out of your way to make hydroxyproline if you're a human, because we have to oxidize proline, and you need all these specific enzymes, you need to put energy into it. Why not just use proline?
I'm not really too sure because they're almost the exact same, except hydroxyproline just has the hydroxyl group on it. I can't really exactly tell you why. It's because they're naturally occurring. I'm not sure why one would have the hydroxy group on it and why one wouldn't.
Okay, well, what do you think about, let's say the entire peptide was all glycine and proline.
All glycine.
So no hydroxyproline, just essentially collagen being glycine and proline.
All right.
Okay. What do you think about how well your cells can handle that protein? So what makes a protein? So if we're talking, because you guys also talked about plastics and polymers, so maybe I'll ask this question to Luke and then come back to you, Michael. Luke, what makes a protein or a polymer, because you had a lot of polymer science in your stuff, processable? Like like we can make a polymer out of basically anything. You can just polymerize anything. But if let's say I make a polymer synthetically, is it enough just to make it, or do I need to be able to do stuff with it? And if I need to be able to do stuff with it, what kind of properties does a polymer need to have so I can handle it and manipulate it?
I feel like, yeah. So I think, I guess I'm guessing you're sort of pushing towards maybe like in like hydrogen bonding. Possibly.
Sure. That so hydrogen bonding helps organize the protein, like polymers and things. But you have like polymers like so polyglycolic acid. I think you both talked about it, and we had an example we were just talking about on a previous slide. So if you, I'm trying to think about how to ask this question carefully. Well, you know what? I'm not sure how to ask this. It's too early in the morning. I'm not thinking clearly on how to best phrase this question. Socratic method. So what I'm aiming towards is solubility issues. So you always, whenever you make a polymer, you need to be able to dissolve it in something. Because if you can't dissolve it in anything, it just precipitates out, and you get what's called basically brick dust. Like it dissolves in nothing, and you have powder, and you can't do anything to it. You can't shape it. You can't process it. You can't treat it with stuff. And that's actually true within the body, except the body's really screwed because as a synthetic chemist, I can pick up my bottle of hexane or my bottle of DMF or my bottle of DMSO or trichlorobenzene at 300 degrees. Like I have lots of options on how to process materials. The body has water at 37 degrees. That's it. That's the only solvent it's got. So what do you think about the solubility of collagen if it was entirely proline and glycine?
If it was entirely proline and glycine, wouldn't that mean the body wouldn't be able to break it down?
I think the body would still be able to break it down. But what do you think of the polarity of the polymer? Like look at the structure of proline and glycine and in a polymer.
Would it just be like non-polar?
Yeah, it'd be really, really non-polar, right? So how soluble in water do you think that would be?
Very not soluble.
Right, and it would actually start aggregating massively. You'd just get these huge clumps of it. So like Alzheimer's disease arises because of misfolding of tau protein, and tau and beta amyloid, and both of those are highly hydrophobic proteins. If they're on their own, they're okay, but if they start misfolding, they become more hydrophobic, and then they start aggregating, and so you would just get these huge aggregates of these things in the cells as they're generating them. Like collagen is pretty non-polar. We don't want it to dissolve, right? I don't want my hair to dissolve in water, or my joints to dissolve in water. Like keratin, sorry for the hair, but collagen, I don't want my joints to dissolve in water. But I also want them as they're growing to be processable before they start kind of assembling into collagen networks. So the hydroxyproline is really important to keep this soluble as it's being made, and it also helps because you've actually shown this down there below in your rope. Like it does make this triple helix, but then the triple helices interact one with another. It's not like one triple helix in isolation. They all kind of intertwine and form these, like they go next to each other, kind of like in your rope image. And hydroxyproline helps with hydrogen bonding to help form that as well. Cool. Again, opening it, if any of the grad students have thought of any questions.
I have one.
Awesome.
There's a triple, right? Triple helix in, we can see in this picture. And we have alpha, I'm sorry, maybe it's not totally related, but there's something in my mind. There's an alpha helix in the structure of protein. Yeah, or just
alpha structure for which one?
Alpha helix. There's an alpha. We have somehow the same things for the in the structure of alpha helix in protein, right? Maybe you should not answer my question. Maybe.
What is your question? What is your question, Rodas?
The alpha helix in the structure of protein.
And how does that relate to the triple helix in this structure?
No, maybe there's not related. I said at first that I had the same, I have a question that I just want to know about the alpha helix structure, that when I saw the triple helix in this picture, something came into my mind about the alpha helix structure of protein. I just want to know more about it. that if you could
So
explain
know
it.
more about alpha helices in general, or how they relate to the triple helix?
If you could, if there's any relation between these two.
Okay,
then
let's
we're
stick with
not-
that as a question. I think asking them for a conversation on the alpha helix would be inappropriate. It's outside the scope of their presentation. Okay, yeah. So if we reduce that question down to how does the alpha helix secondary structure of proteins relate to the triple helix structure?
Yeah, maybe there, if there's any relation, or if they have any same, I mean,
I absolutely, yeah.
application. I mean, I don't know.
So is the question just how does the alpha helix relate to the triple helix?
Is there any relationship, and what do you want to discuss, like the differences or differences in level of the interactions?
I'm not really too sure how they interact, but I know that every polypeptide chain in this collagen is twisted the same way, and the helix coils in the opposite way, so that it can be structurally strong. It's kind of hard to visualize here because they made it so many colors, but I think that's generally what's going on there. That's why I compared it to the rope. Like, this is also a very bad quality image when it's blown up, but there's like the strands that are twisted into the actual rope, and then those ropes themselves are twisted in the opposite direction, and it makes it stronger. I don't know if anyone's like braided hair either. I had long hair for so many years, but like when you twist it in one direction and then coil it the opposite, it'll stay together because it's just a strong interaction.
So I guess maybe so we didn't spend a lot of time talking about some of the basic protein stuff, but there's primary, secondary, tertiary, and quaternary structures for proteins. Does that ring a bell?
Yeah, it does.
So an alpha helix is what kind of structure?
An alpha helix, that's a secondary structure, right?
Right. And what's your triple helix?
I think that's a tertiary structure.
Is it the three-dimensional shape of an individual protein?
Three-dimensional shape
of an individual protein?
I would say yes.
What's the definition of a quaternary structure?
Isn't it made out of multiple tertiary structures?
Yeah. So multiple different proteins coming together to make a quaternary structure. So the shape of each, so the tertiary structure of each member of the protein affects the tertiary structure of the other members of the protein to create a new emergent sort of three-dimensional shape.
So is it technically a quaternary structure?
Yeah, because you've got three different proteins, right? Each of these collagen chains is different. They're not the same molecule. They're three different collagen chains. So there's three different molecules coming together to make a new shape. So do you think an individual collagen chain on its own would make a helix necessarily? Like the same kind of helix as this?
Not a triple helix. It'll be an alpha helix. Or is that the term for it?
I think it would probably be a polyproline type 2 helix because it'd be so rich in polyproline, or in proline. But it's not quite an alpha helix. But the details are silly. Angle differences and Ramachandran plots and stuff, but it doesn't matter. Yeah, okay, cool. So this is a quaternary structure rather than a secondary structure. I think that's a good answer. Any other questions? Any questions in the chat? Cool, okay, Luke, if we go to your presentation. Good job, Michael. I'll just share your screen. If you want to go to just some of the places where you're talking about the synthesis of, so it's a cool presentation. I love this stuff. That's actually, well, I got a question about that, but I'll come back to that because it's a little tougher. Let's go back earlier. I want to go back to your synthesis of your block copolymers. Here, cool. Did you look into how these kind of block copolymers are synthesized?
Most of the papers I looked at kind of had these already pre-made. They barely even really went into the polymerization, but not really, no.
So there are a couple ways of doing this. How would you and if you're sort of given this, how would you think about trying to polymerize this? How would you think about making a diblock copolymer of isoleucine and lysine, or leucine and lysine?
How would I go about making the
Yeah,
individual?
so an individual chain, and they'll self-assemble, right? So you need a block copolymer, you need all leucines, and then you need all lysines. So what kind of strategy would you use?
I'm not 100% sure how to approach that.
So later on in your presentation, I think you discussed some ways that people can make, like chemically make peptide polymers. Like, so what you're doing here, these are all chemical syntheses. These are not biological syntheses, right? You're not taking a ribosome and dumping in some mRNA that says leucine, 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 and a bunch of lysines. So what were some of the ways that you talked later on in the presentation about synthesizing these kinds of things?
I'm not quite sure what you're.
So you have some syntheses. So that's all self-assembly. Keep going. Keep going. Keep going. Keep going. There we go. Okay. So you could use a chemoenzymatic synthesis. So with a standard chemo, so if I just took a whole lot of amino acid esters, dumped them in with the protease, like a standard non-specific protease, what kind do you think I would get, like a random polymer of these things? Like if I have like 10 phenylalanines, 10 tyrosines, 10 serines, and I toss them all in like into a soup and I throw in some protease, what do you think about the polymer I'm going to get out of there?
I think it would be sort of a random.
Yeah, I agree. I agree. You just, within reason, there'll be maybe slight differences. Those deviate slightly from randomness, but it'll be basically random. Okay, how could I manipulate this to make that block copolymer experimentally? So, set you up. You got a round bottom full of protease. You got a bucket of leucine ester and lysine ester, and another bucket of lysine ester. But you wanted an all leucine and all lysine block, more or less. How would you go about doing this?
Could you just make the blocks individually at first?
Yeah, you can make them individually, but you can actually do can you think of how you could do it? So you can make them individually, then couple them together using something like native chemical ligation or click chemistry. And we can actually, we're probably not going to get around discussing it today. I think that this will be the end of the day. But if we're, there's actually a way you can make them in one pot, like in one reaction, not having to do any kind of workup.
Yeah, I'm not sure how. I'm also thinking about how it would work hydrophilically, since we have one of these. One of these was
Hydrophobic, one was hydrophilic.
Yeah.
So the protease, you'd probably actually end up using, some of these can be very functional in an organic solvent, which is nice. So you can often use a mixture of like THF water, which can dissolve everything, basically. So let's say solubility is not a concern. I think that is really valid, depending on how nonpolar your nonpolar thing is. You can get into serious trouble over solubility, and that is actually a major concern. So you're absolutely right. But let's say that isn't a concern. Let's say solubility is not an issue. So you got a bucket of protease, in a flask with your magic solvent mixture that's going to dissolve everything. You got a bunch of lysine esters, and you have a bunch of leucine esters, and you want to make a diblock copolymer.
I feel like I'm so close, but yet so far. Hmm. I'm sorry, I'm missing something here.
Well, you're pretty. So what happens if you dump in all the leucine esters? Okay, and you let it go for 24 hours. What will you have in your flask? So you take your protease, you have your protease flask, you dump in all your leucine esters, and you let it go for 24 hours. What are you going to have?
I would say a big glob of leucine polymer.
Yeah, you'd have a whole bunch of leucine polymer chains. Would you have any leucine monomer left?
I wouldn't think so, no.
I disagree. I think you'd have some.
Okay.
Things never go to completion, but you'd have some. Okay, now what if I take, so what do you have at the terminus of your polymer? Is it still, basically, is your polymer terminated or is it still a living polymer? Can I still add to it?
I guess it could still be active.
Yeah, because there's no reason it wouldn't be,
Yeah.
right? Like the polymerization is over because you're running out of monomer, not because you've terminated your polymer. So you have this leucine polymer now. What if I now take your lysine ester and I dump it into that flask?
Yeah, then I guess that would create the copolymer.
Yeah, you'd start adding lysine to the leucine.
Okay. All right. I guess, is that it?
Yeah, that is how you.
All right,
That is
that's
literally
simple
how
enough.
you do this. You can do it chemically, you can do it chemoenzymatically, but that is literally how you make diblock copolymers. You just time it. You build one block of the polymer, then you add the next reagent and build the other block of the polymer. Often you get tri-block polymers that way though, because you can add to both ends of the polymer. There's nothing stopping lysines adding to the C terminus or the N terminus of your leucine polymer. So you might get a tri-block copolymer. That's not a problem because what just happens is your lysines would, your hydrophobic polymer would sit in the middle and your hydrophilic polymers would kind of loop out both to the outside. If we go back to your mechanism that you have on like your second last slide, is the yeah, cool, that one. So this is really, really, really cool. I agree with you. So what you wise, oh no, you didn't. I was thinking you wisely left something off your slide, but you didn't. You said commonly used for its molecular weight control. How does, and you sort of said, hey, this is a good idea, this is a good strategy of this, it really controls molecular weight. How does this control molecular weight? Because the idea is like a condensation polymer. So, I take nylon, or I take, the cyclic lactam that is a precursor for nylon. If you're making it that way, I can't remember what it's called, but I've done this. You add a little bit of a nucleophile, you heat the crap out of it, and it starts ring opening polymerizations, right? Like you were showing the ring opening polymerizations. And you don't have you don't really have any control over dispersity. Like some polymer chains will grow really large, some will grow small, it's going to be kind of random. This gives you a much tighter polymer dispersity. Most of the polymers are about the same length. So how does that work?
I want to say it's because the next addition of monomer can't be carried out without the previous one transferring the silicon group. Am I on the right track with that? Sort of how it's the next step is determined on the previous step finishing. Maybe that just more control that way. It's easier to terminate in that aspect.
Well, if one reaction is gone and it's carried, one molecule is carried through, so what that would matter is if you get control of the polymer length, if the starting material reacts faster than the product of each addition. Because then you're slowing, if you want to do it that way, if you want to sort of control it that way, the way you're saying, then you control it because the starting material reacts faster than product. And so everything kind of slows down over the reaction so that everything kind of catches up. Otherwise, like it's true, you can't do until that, but now you have your product on the bottom left-hand side and you have your starting material on the top left-hand side. And to me, both of those look ready for the next reaction. Like both those are ready to add the nucleophile and do the next Michael addition equally. Do you agree or disagree?
Yeah, I would agree.
Okay. So if it's not that we're getting product inhibition, which is that, like the product reacts slower, any kinetic, so if we talk about kinetic versus thermodynamic processes, like any kinetic process is irreversible, right? So if your polymer chains start all growing, then you're kind of fucked with polymer dispersity. But if you're under, let's say you have something that's completely under thermodynamic control, like a reaction, so a reaction is completely under thermodynamic control. Do you think that polymerization to thermodynamic control is going to have a better polymer dispersity than a polymerization under kinetic control? And why?
I would think. I want to say it would be better under thermodynamic
You're
control.
absolutely right. And now think about what that means and why it would be better. So I think the big question I've got for you here is the energy of the starting material and the product, the top left and the bottom left structures, are those the same? Do you think those would have the same stability, thermodynamic stability? Or would they might not be massively different, or would they be different?
I would think that the bottom left would be more thermodynamically stable. Like,
I think up to a point, I agree with you, but we'll agree that they're different.
yes.
If they're, so what ends up happening is okay, and then let's say this was under thermodynamic control. What would need to be necessary for something to be under thermodynamic control?
I would just say, and like enough energy to get the most stable product.
Okay, I completely agree. What is a requirement of all thermodynamically controlled reactions?
I'm not quite sure.
They have to be in equilibrium.
Okay, yeah.
So if this reaction was all in equilibrium, it will be under thermodynamic control, and that will give us dispersity control. That's not the case here. This is not in equilibrium. Okay. What's the slowest step of this reaction, you think?
Let's say the Michael addition.
I don't agree.
No? Okay. Would it be the nucleophile?
Nope, that's gonna be fast.
Oh yeah.
You've only got one left.
Yeah, so I guess it's
That one.
Yeah.
That MMA, that rearrangement.
Yes.
So that rearrangement, is the starting material reactive?
This this top right here.
Right. So can that do another Michael addition?
Not.
I like how you were saying that.
Yeah, I don't think so.
I would say not easily.
Yeah.
It can, is nothing stopping it, but it's not easy as a nucleophile. So what it wants to do is it wants to rearrange to that bottom product, which can then do another Michael addition. Okay? So that step, that rearrangement of shuffling the methyl group, so what ends up happening, right, is you regenerate the double bond, the rings flip, you regenerate the double bond the other way. You have to sort of interconvert. That reaction is slow. And so what that means is what will end up happening is once you add a certain amount of nucleophile, however much nucleophile you add, you'll attack that many silicons. They will all do the micro additions. They will all make the top right structure. Then they will slowly migrate to the bottom right structure. But by the time they migrate to the bottom right structure, all of the nucleophile activated silicon groups have done the micro addition. So you have as many chains growing as you added nucleophile, more or less. And then they do the next micro addition. Each one of those can then do the next micro addition. So none of them really get far ahead of the others because the time it takes for them to do that rearrangement, they've all done a micro addition already. So everybody can kind of catch up at that step. So you don't sort of have one polymer chain racing ahead of all the other polymer chains, getting longer, really fast. And there's a bunch of different polymerizations that are based on this principle, that they have a really slow step involved in them, which slows them all down.
Okay,
so
that's interesting.
Yeah, it's a cool way to control length. So all the chains tend to be about the same length because you don't kind of get the rich getting richer with one polymer chain getting a whole lot faster than all the others.
Yeah, it makes sense.
Which is what sometimes happens with condensation polymers. Okay, cool. Last call for questions. Got something in the chat here. Ronaz is thanking Michael. Okay. I think that's it. We're at 9:50. Good job, guys. Good presentations. If you want to submit me the PowerPoints with the references in there, I'll use that as the basis for the marking. If you want to put the references in there, I'd strongly recommend, I think I pointed out in the syllabus, but I strongly recommend you use the American Chemical Society format. You're free to use like the Royal Society of Chemistry or the Wiley format if you prefer, but just be consistent if you use one. And no need to put the titles in the references, just the citation is fine. You don't have enough room for titles, they take up all the space. So yeah, that's it. I will see everybody Wednesday. Good job, guys. Those are good presentations.
Thank you very much.
Okay.
So we're now gonna sort out the final date.
Oh yeah, shit. Yeah, we'll sort out the final. Fuck, good point, Hazel. Okay, I'm happy with the, so those of you, I think Apurvi and Mohammed and Rona has joined us after we were talking about this. We can have the final any date between December 10th and December 22nd. At any time. What works, so what I'm going to favor is I favor the MMB students and the undergraduate students because they have other exams. So if some people want to throw out some dates, we can just start discussing it.
I was convinced that the final was on the 21st, and I'm leaving for the holidays. So that's when I planned on taking it.
Okay, so 21st is fine. So no later than that.
Yeah, I was good with the posted date as well.
Oh, is that posted?
It's posted on the UN site, but the date posted would work well if my
it was December 21st. Do you know what the time was?
7 at night.
Okay, let's not do 7 at night. Do you want noon?
Yeah, that would work for me if it works for everybody else.
That works for me.
21st.
Sorry, noon on the 21st?
Can we do like even one day earlier than that? Because I might be on my way to Toronto on 21st. And very possibly on my way to Toronto on 21st. But I'm not favored, so just an idea. It's actually okay.
If there is a posted date, then I kind of need near unanimity to override it. Luke or Michael, do either of you guys have trouble moving it to the 20th, or you prefer on the 21st?
No, I have no problem with that.
I do have an exam at the 20th, but I mean,
okay, 21st. I think it stays the 21st then. Sorry, Hazel, you might need to write the exam on the train and hope you have connection.
If it's new, it should be fine.
We can move it a little bit earlier if you like.
It's Sorry, what?
We can move it a little bit earlier if you like.
I really don't know because the plan is actually I'm gonna take Mike's car and I actually like it's not set anyways. So
Okay, so maybe let's leave it for noon on the, does that work for the MMB students? 21st at noon.
Yes, it works for me. It works for me.
Works for you? Okay, good.
Yes.
I wasn't aware there was a posted exam date. Okay, not hearing any disagreement, we'll stick with the 21st. We'll move the exam from 7 p.m. to noon so that it's not a night exam. And I don't know how many of you guys are overseas right now, but if you want accommodation based on time zones, let me know and I could arrange something. If not, if absolutely necessary, try and avoid it because you will get a different exam. I don't want to write 2 exams. Okay. See you guys Wednesday for the last class where we're going to discuss click chemistry and some bioconjugation stuff. Take care.